everybody, I'm back. My name is Elisa, and we are talking about male reproductive system, and we're going to continue on, with some more on sperm production. Sperm production, primordial germ cells, right, are going to form in the yolk sac of an embryo. And so what ends up happening is they're going to colonize these gonadal ridges and become spermatogonia. Puberty brings on the actual manufacture of sperm, known as spermatogenesis, right? So the beginning of a sperm. Spermatogonia lie along the periphery of the nursery, known as the seminiferous tubules, and then they begin to divide by mitosis. They also go through meiosis later on. We'll talk about that. One daughter cell of each one of those division is going to stay in the tubule wall because it's like going to be kind of a stem cell. We're going to use it over and over and over and over and over. And we call this the type A spermatogonium. Okay, they're like just kind of holding down the fort. The other ones, they're going to go on to become full-blown sperm, right? It needs to migrate away. We call these the type B. That's the other division half. And so they're going to begin this really cool journey. And so let's talk about that. So spermatogenesis, right? So the type B spermatogonium is going to get bigger, right? And it becomes eventually a primary spermatocyte. And this is where crossover happens, right? And some of those important points of meiosis. Nurse cells are going to engage at this point because it's the nursery, right? So you gotta have nurse cells. They're going to protect it from things like the immune system for sure. They're going to provide it with waste removal, with nutrients, with anything it might need. They're going to form the blood testes barrier, the BTB, which is like the blood-brain barrier, and just kind of keeps that immune system at bay. That primary spermatocyte is undergoing meiosis 1 right here during crossover, which is going to give rise to two equal size, eventually, equal size haploids, and these are going to be genetically unique secondary spermatocytes. And each one of these secondary spermatocytes is going to go another round of cell division, right? And they are actually going to become haploid, right? So here we go, right? And so that whole point is just because, we're cutting down from like 2N to just N and N, right? Just cutting that genome in half. So that's round 2. And so we should end up with 4 spermatids that are about ready to get going into becoming mature sperm. So they're going to start their differentiation into the spermatozoa. So they can go from being just, sacs of chromosomes into like super fast swimming sacs and a bunch of other stuff of chromosomes. Sperm production. So when a primary spermatocyte undergoes meiosis, it becomes genetically different and needs to be protected from the immune system because of that crossover, right? Crossover is what did it. Primary spermatocyte is going to move towards the lumen, right? The lumen is like that inner part, right, of the seminiferous tubule, so it can travel, it can transport. And then there's going to be a new tight junction between the nurse cell that's going to form behind it, kind of closing it off as it goes. So that's how we create that blood testes barrier. This is really cool. I like this one. So this is like more, I don't want to say the evolution, but the maturation, right, for the most part. So here is a spermatid, and it's going to start kind of, we needed to get to this like race car version of a cell, incredibly powerful cell. And so what we are going to eventually have is an acrosome, and that's going to help us like with enzymes and different types of biological chemicals that are going to allow us to kind of like penetrate into the egg cell itself. The nucleus is going to hold the DNA, so all the goods. Basal body is connecting, and so we'll talk a little bit about that and how that works later. But like right in here is this like piece of the tail, which is chock full of mitochondria, right? And so it needs a lot of it because of the way that is going to like spin the cell. It's like kind of does this like spinning motion, the flagella, and that's how it zooms across in the media. And so this is just like the development of those components, right? Like here's the acrosome developing here, right? Like the shrinking up here of the nucleus body itself. Basal body remaining connecting, so developing mitochondria, centering them around that middle point of the tail. And then little bits of shaping going on here, like cytoplasm is kind of like being removed, remodeled. A spermatozoid has two parts, a head and a tail, and the head shapes like a pear. And then other stuff here. Okay. The nucleus is going to contain that haploid set of chromosomes, right? And the acrosome up here at the top is going to have that cap over the apical half of the nucleus. It's going to have the enzymes that are going to help us penetrate the egg. The basal body at the basal end is going to be like what attaches the flagella onto basically the component that holds the nucleus. The tail is divided into three major regions. Okay, so we have, here's the sperm. Okay, three regions. Okay. The middle piece is going to contain the mitochondria. They're going to be right here. And around an axoneme, the flagella. And so that's going to produce all that ATP because in order for it to spin and spin and spin, we're going to need to pay it off with the energy currency of the cell. The principal piece of the axoneme is covered by this sheath and supporting fibers, and it constitutes most of the tail, which really, if I had a picture of how it looked, like it's so amazing in there. Like there's, oh, that's terrible. You have to think of this like amazingly complex mechanism. Very cool stuff. Anyway, so the end piece is the very narrow tip end of the flagella. Okay, that's it, I guess. So, well, I hope that was helpful, actually. I hope it was helpful. I think it's interesting. And so I hope you're out there somewhere having a good day. Poor little, maybe I'll give this happy face some hair. There we go. Yeah, a little bit better now. All right, I hope you're having a great day. Bye-bye.